Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore a fascinating hidden property of the molecules that make up your body. It is a geometric secret that determines whether a medicine will cure you or harm you, and whether your body can extract energy from the food you eat. Let us dive into the world of chiral centers and polarized light. To begin our journey, imagine you are trying to put your right glove onto your left hand. No matter how much you turn, twist, or stretch that glove, it simply will not fit properly. Your hands are perfect mirror images of each other, yet they cannot be perfectly aligned on top of one another. In the world of chemistry and biology, molecules can possess this exact same property of handedness, which scientists call chirality. Just like your hands, these molecules exist in two mirror image forms that cannot be superimposed. While this might seem like a tiny geometric detail, in the microscopic world of your body, this shape difference changes absolutely everything. Now that we understand what molecular handedness is, let us look at where it comes from. In biochemistry, a chiral center is typically a carbon atom that is bonded to four completely different chemical groups. Because carbon forms a three-dimensional tetrahedral shape, attaching four distinct groups means the molecule lacks any internal plane of symmetry. If you were to draw a line right through the middle of the molecule, the two halves would never look identical. To identify a chiral center, you simply look at a carbon atom and ask yourself if all four attached branches are unique. If they are, you have found a chiral center. Building on the idea of the chiral center, we find that when a molecule has one, it can exist as two distinct stereoisomers. We call these enantiomers, which are simply non-superimposable mirror images of each other. Think of them as the left-handed and right-handed versions of the exact same molecule. However, if even two of the attached groups on the carbon atom are the same, the molecule loses this property. It becomes achiral, meaning it has a plane of symmetry and can easily be superimposed on its own mirror image. Recognizing this difference is the first step in understanding how molecules interact in three-dimensional space. You might be wondering why this molecular handedness matters so much in real life. The answer is that almost all the biological machinery in your body, including enzymes and receptors, is highly sensitive to chirality. Your body is essentially a collection of molecular locks, and only the correctly handed key will fit into them. For example, your body can easily digest D-glucose for energy, but it cannot process its mirror image, L-glucose, at all. A tragic historical example of this is the drug thalidomide, where one mirror image form safely cured morning sickness in pregnant women, but the other form caused severe birth defects. This is why understanding chiral centers is absolutely critical for designing safe medicines. Since enantiomers are so different biologically, we need a way to tell them apart in the lab. Because they have the exact same physical properties like boiling point and density, standard physical tests fail us. However, they interact differently with something called plane polarized light. Normal light waves vibrate in all possible directions, but when passed through a special filter, the light waves vibrate in only one single flat plane. When this plane polarized light passes through a solution containing chiral molecules, the molecules actually twist or rotate the plane of the light. This twisting effect is our key to measuring chirality. Before we look at the math, let us visualize this process with a diagram of a polarimeter. On the far left, you see unpolarized light vibrating in all directions. It then passes through a vertical polarizing filter, emerging as plain polarized light with arrows pointing only up and down. This flat wave then enters the cylindrical chiral sample tube in the center. As the light travels through the chiral molecules inside the tube, notice how the plane of the wave gradually tilts. Finally, on the right, the light emerges rotated by a specific angle, which we call the observed rotation, alpha. This diagram perfectly captures how we detect molecular handedness. Now that we have seen the polarimeter, let us understand the mathematical formula we use to measure this twisting effect. We calculate a standardized value called specific rotation, denoted by alpha in square brackets. This standardizes the measurement so scientists worldwide can compare results. In this formula, the regular alpha is the observed rotation, which is the actual angle in degrees that the light was twisted. The variable C represents the concentration of the chiral substance in grams per milliliter. Finally, L represents the path length of the sample tube in decimeters. If you increase the concentration or the tube length, the observed rotation will increase because the light hits more molecules, but the specific rotation constant will always remain exactly the same. Let us put this formula to work with a concrete example. Suppose you dissolve 2 grams of an unknown sugar in enough water to make 10 milliliters of solution. You place this in a 1 decimeter tube and observe a rotation of positive 1.5 degrees. First, we must find the concentration by dividing the mass by the volume, giving us 0.20 grams per milliliter. Next, we substitute our observed rotation, concentration, and path length into the specific rotation formula. Dividing positive 1.5 by the product of 0.20 and 1 gives us a final specific rotation of positive 7.5 degrees. The positive sign tells us this molecule is dextrorotatory, meaning it rotates light to the right. Having successfully used the formula to find specific rotation, we can also use algebraic manipulation to solve for an unknown concentration. This is a very common and powerful tool in biochemistry laboratories. Let us set up the derivation. We start with our standard specific rotation equation. Our goal is to isolate the concentration variable, C. To begin moving variables around, we first multiply both sides of the equation by C. This brings the concentration out of the denominator and places it on the left side, multiplied by the specific rotation. This intermediate step sets us up perfectly to isolate the concentration completely. Continuing from our previous step, we now have the concentration multiplied by the specific rotation on the left side. To isolate the concentration completely, we simply divide both sides of the equation by the specific rotation. This gives us our final rearranged formula. Concentration equals the observed rotation divided by the product of the specific rotation and the path length. This rearranged formula is incredibly useful. It saves us from having to evaporate the water and weigh the sticky sugar residue just to find out how much substance is dissolved in a solution. We can find the concentration just by shining light through it. Now that we have derived this new form of the equation, let us use it to find the concentration of an unknown D-glucose solution. We know the specific rotation of D-glucose is positive 52.7 degrees. We measure an observed rotation of positive 10.54 degrees in a 2 decimeter tube. We plug these values into our rearranged formula. On the bottom, we multiply the specific rotation by the path length of 2 decimeters. Dividing the observed rotation by this denominator gives us a final concentration of 0.10 grams per milliliter. This means there is exactly 1/10 of a gram of glucose dissolved in every single milliliter of the solution. We have covered a lot of ground today, so let us review the key takeaways. First, a chiral center is usually a carbon atom bonded to 4 unique groups, giving the molecule a specific handedness. Second, because biological systems are highly sensitive to shape, this handedness determines how a molecule functions in the body, like a key fitting into a lock. Finally, while we cannot distinguish these mirror image molecules by sight, we can identify them by how they rotate plain polarized light. By measuring this optical rotation, biochemists can easily determine the identity, purity, and concentration of vital biomolecules. You now understand the fundamental principles of molecular chirality.